The popular story makes dog domestication sound almost simple. Ancient humans found a wolf pup, raised it by the fire, and slowly turned it into a dog. But mitochondrial DNA sequencing and archaeological layers tell a different story. Multiple genomic studies trace the wolf-dog split back 20 to 40,000 years, long before agriculture began 12,000 years ago, long before humans ever truly settled down. Picture the Ice Age. Humans could barely feed themselves. Who's hand-taming an 88-pound apex predator with a mouthful of teeth? The thermodynamics don't work. The survival logic doesn't work. One leading possibility is far stranger. Domestication may not have begun with humans choosing wolves at all. Some wolves may have hacked their own way into our world. So if humans didn't deliberately invite them in, how did these prehistoric killers walk straight into our camps? No ropes, no cages, nobody forcing them. The answer may have started with trash. Late Pleistocene human camps were ringed with leftover bones, guts, and high-calorie scraps. Free food, zero hunting cost. But not every wolf dared approach. There's a variable called flight distance, how close a threat can get before an animal bolts. Wolves with naturally lower fear and stress responses may have had shorter flight distances. They were bolder. They ate first, ate more reliably, and were more likely to out-reproduce the rest. This wasn't survival of the fiercest anymore. It was survival of the friendliest. The anxious, aggressive wolves were less successful around human camps. The calm ones survived, and passed those behavioral tendencies to the next generation. Getting less aggressive to raid a trash pile, sure, that tracks. But why would a personality shift completely rewrite their looks? Is friendliness actually a physical shape? In 1959, Soviet geneticist Dmitry Belyaev launched a wild experiment in Siberia. He selected for exactly one trait: tameness. Each generation, only the calmest silver foxes, the ones most eager to approach their handlers, got to breed. 20 to 30 generations later, something strange happened. The foxes weren't just acting like dogs, they were starting to look like them. Floppy ears, curly tails, patches of white fur. Nobody selected for those features. They just showed up, riding shotgun with the tameness genes. One influential hypothesis traces this pattern back to early embryo development. Changes involving neural crest cells, cells involved in the stress response, pigmentation, cartilage, and other tissues, may help explain why selecting for behavior can produce unexpected physical traits. Select for personality, and you may accidentally change far more than behavior alone. That genetic chain reaction may help explain why they started looking like dogs. But once humans went agricultural and swapped meat for grain, how did these born carnivores even survive? Genomics has the answer. Wolves generally carry fewer copies of AMY2B, a gene involved in producing pancreatic amylase, an enzyme that helps break down starch. Many modern dogs carry substantially more copies, although the exact number varies widely between breeds and populations. That pattern suggests that, as dogs lived alongside farming societies, natural selection increasingly favored animals better able to digest starch-rich human food. As meat became less reliable and grain scraps piled up, dogs that happened to carry extra amylase copies gained a real survival edge. Environmental pressure gradually pushed their digestive systems away from the wolf baseline, helping turn specialized hunters into opportunistic scavengers that could live off human leftovers. Digesting our leftovers kept them alive. But to get humans to willingly hand over precious food, they needed something more powerful. That advantage may have been a new kind of cross-species emotional connection. A 2015 study in Science found that when dogs and their owners maintained eye contact, oxytocin levels increased in both. That's the so-called love hormone, a chemical involved in social bonding, trust, caregiving, and attachment. The researchers tested hand-raised wolves too. Even wolves raised by humans from birth couldn't trigger the same oxytocin spike through eye contact. Dogs appear to have developed an unusual ability to enter a hormonal feedback loop with humans using nothing more than sustained eye contact. Your brain may not literally mistake your dog for a child, but it can activate some of the same bonding chemistry. Eye contact alone wasn't enough. To strengthen that emotional connection, dog evolution may even have reshaped the face itself. A 2019 anatomical study in the Proceedings of the National Academy of Sciences uncovered something remarkable. Researchers dissected and compared facial muscles in wolves and dogs and found dogs carry a muscle wolves either completely lack or have only in vestigial form, the levator anguli oculi medialis, or LAOM. This muscle raises the inner eyebrow, making the eyes appear larger and producing an expression humans often interpret as vulnerable, attentive, or childlike. Researchers believe this muscle was likely shaped at least partly by human emotional preference over our long shared history. What looks like ordinary begging may be the result of generations of dogs benefiting from expressions that humans found difficult to ignore. They rewired their digestion, their hormones, their facial muscles. But can they actually read our minds? Cognitive scientists Brian Hare and Michael Tomasello designed a classic experiment, the pointing gesture test. Hide food under one of two identical containers, then point at the correct one and see if the animal follows the cue. The results were striking. Young puppies often followed the human gesture with little formal training, and under comparable conditions, they tended to use human social cues more readily and consistently than wolf pups. Even chimpanzees can struggle with this kind of cooperative pointing task. Experience still matters, but research suggests that domestication gave dogs a strong inherited readiness to pay attention to human communication. After tens of thousands of years of selection, nature had built humanity a near-perfect companion. But selection did not stop. Its goal changed. Once humans realized they could control which dogs reproduced, survival was no longer the only force shaping the species. A centuries-long experiment in deliberate genetic reshuffling was about to begin. From this point on, dog evolution answered to human will, not nature. Archaeological bone analysis shows ancient Egyptians, over 4,000 years ago, bred lean, long-limbed sighthounds built for high-speed pursuit, strikingly close to today's Saluki. At almost the same time, in Mesopotamia, humans pushed the opposite direction, breeding massive, heavy-boned mastiffs for war and guarding livestock. Working with an extremely limited founding gene pool, humans molded the same species like clay, stretching and compressing its skeleton toward two opposite extremes in just a few dozen generations, all to serve hunting speed, military defense, or herd protection. Reshaping bodies for real survival needs that still made biological sense. But once humans stopped needing dogs to hunt or fight, breeding motives took a far darker turn. Mid-19th century England. The Kennel Club's founding in 1873 marked the rise of dog show culture, and with it, an entirely new judging system, the breed standard, a written blueprint dictating ear shape, coat color, body proportions, down to the inch. From here on, breeding wasn't about whether a dog could work, hunt, or guard. It was about whether it matched the blueprint. Judging criteria slid completely from function into an extreme aesthetic competition. At the same time, the closed stud book system took hold. Once a breed was officially defined, it could only be bred within that sealed gene pool, permanently cutting off any chance of bringing in outside genes to restore diversity. To maintain that artificially defined purebred look, humans turned to a breeding method that could preserve appearance while quietly concentrating genetic risk. That method is systematic inbreeding. The coefficient of inbreeding, COI, measured through pedigrees or genome sequencing, can be alarming. Multiple genetic studies on modern purebreds show that some breeds have average levels of genomic relatedness comparable to extremely close family matings. In breeds such as the pug, a relatively narrow founding population and generations of closed breeding have left limited genetic diversity. This isn't a thriving genetic system. It's a shrinking pool recycling many of the same genes generation after generation. Diversity drained away, while harmful mutations that might once have remained rare become amplified and concentrated. This genetic purification spiral has produced brutal consequences. And nowhere is it more direct, more merciless than the structural damage done to their skulls. You've seen the flat-faced dogs, English bulldogs, pugs, French bulldogs. Vets call them brachycephalic breeds, and skull x-rays make the problem obvious. Chasing an ever flatter face compressed their skull length, but the soft tissue inside, the soft palate, the tongue, barely shrank at all. The result? An oversized soft palate and a dangerously narrow airway. This is the clinical condition known as brachycephalic obstructive airway syndrome, or BOAS, and veterinary studies show that many individuals in severely affected breeds suffer real breathing difficulty. That adorable snoring and wheezing everyone finds so charming? Medically, it's a lifelong, chronic struggle to breathe. Some can't even finish a short walk without gasping. Wrecking their airways wasn't enough. Human aesthetics also destroyed the skeleton that's supposed to hold their bodies up. The dachshund's body is basically a living experiment in extreme proportions. Bred originally to burrow after badgers, they got an absurdly elongated spine and stubby legs. But that overextended spinal column dumps far more shear and compressive force on every single disc than it should. That's exactly why veterinary data shows dachshunds have some of the highest rates of intervertebral disc disease, or IVDD, of any breed. Now look at some modern showline German Shepherds. In the pursuit of a dramatic sloped top line and exaggerated rear angulation, breeders may compromise natural movement and joint stability. These structural choices may contribute to existing risks of hip and mobility problems. If skeletal deformity is cruelty you can see with your eyes, a different genetic flaw hiding beneath the skull is putting some dogs through a lifetime of hidden neurological torment. The Cavalier King Charles Spaniel is the textbook case. They suffer from a condition called syringomyelia. MRI studies show that, in affected dogs, the back of the skull can be too crowded for the brain tissue it contains. There's not enough room in the back of the skull, so brain tissue gets forced down into the upper spinal canal, space that's only supposed to hold the spinal cord. This causes agonizing, chronic nerve pain. Veterinary records show a striking share of affected dogs display a telltale behavior, scratching at their own neck and ears in midair, trying to relieve a deep, unlocatable pain. A symptom so consistent, it's now written into the breed's clinical diagnostic guidelines. By Darwin's own logic, animals that can't give birth naturally or breathe properly should have been wiped out long ago. So why haven't they gone extinct? Why are they instead among the most numerous carnivores on Earth? Because their real habitat isn't the forest anymore. It's the human home, supported by human demand and emotional attachment. In modern society, fitness, Darwin's term for reproductive success, has been completely redefined. A breed that's adorable, but carries a severe congenital heart defect or airway deformity can, thanks to sheer market demand, out-reproduce a genetically healthy but ordinary-looking mutt by a wide margin. They no longer need to survive harsh winters, scarce prey, or predators. They only need to appeal to human taste and emotional consumption. Human protection, money, and emotional preference have become some of the strongest forces determining which dogs reproduce. We caused this genetic disaster with our own hands, and science has known how to fix it for decades. So why has the rescue plan stalled for so long? The scientific fix is actually simple. It's called outcrossing. The most famous case is the LUA Dalmatian project, launched in 1973 by geneticist Robert Shibely. Dalmatians are unusually prone to hyperuricosuriasia, a genetic condition that raises uric acid levels and increases the risk of painful urate stones. His solution? Introduce genes from a single English pointer, then backcross for several generations, eliminating the fatal defect while preserving almost all of the Dalmatian's original look and build. The project demonstrated that a targeted outcross could improve health without erasing the breed's recognizable identity. Instead, it took nearly 4 decades before major kennel clubs would grudgingly grant it official recognition and pedigree registration. Who exactly resisted a scientific fix like this? Why would anyone rather watch an animal suffer than let go of so-called pure bloodlines? The resistance is baked into the system itself. Look at over a century of how major kennel registries actually operate, and the pattern is clear. Dogs stopped being treated as living creatures and became something closer to protected intellectual property, like a trademarked brand, with every pedigree certificate acting as a deed of ownership. Break the bloodline barrier, let outside genes in, and you threaten that brand's exclusivity, its scarcity, and the commercial value built up around it for generations. The problem is therefore not purely scientific. It is also cultural, institutional, and economic, tied to tradition, status, competition, and the value placed on closed bloodlines. If the existing institutions can't save these dogs, can rapid advances in genetic technology finally break us out of this dead end that traditional breeding built? The answer is already unfolding quietly in labs. Commercial canine genetic panels are already giving breeders information that previous generations never had. At the same time, CRISPR and other gene editing tools are being explored in laboratories, although their routine use in dog breeding remains far in the future. Modern genetic testing can use a saliva or blood sample to screen dogs for many known disease-associated variants before breeding happens, rather than waiting until illness appears in the next generation. That creates an opportunity to judge breeding decisions not only by whether a dog matches the visual blueprint, but by measurable health, genetic diversity, temperament, and function. The technology is not perfect. Many common disorders are polygenic, influenced by dozens of genes, body structure, and environment. A DNA test cannot guarantee a healthy dog. But for the first time, breeders have tools that can replace at least part of the guesswork with evidence. We've now got our hands on information that natural selection once tested only through survival and death. So what will the dog of the future actually be? Not today's genetically isolated purebred, not a random street mutt, but something else entirely.
The answer is purpose-bred functional crosses. Carefully planned crosses selected for health, temperament, and performance rather than novelty alone. Leading service dog organizations worldwide, including Guide Dogs UK, are actively abandoning strict purebred lines in favor of precisely planned crosses, like the Labrador Golden Retriever mix. These programs combine health screening, behavioral data, and controlled crossing to produce dogs with reliable temperaments and strong working ability. Greater genetic diversity may also reduce the risk of some recessive inherited disorders, although crossing breeds is not an automatic guarantee of health. This isn't some random pairing on the street anymore. It's an entirely new approach to functional breeding, backed by precise science. 30,000 years, full circle. We've ended up back in the lab, at the exact same starting line nature used to screen those first wolves: health and survival. Let's fast forward through 30,000 years of shared evolution. It starts somewhere in Ice Age Eurasia, with a less fearful wolf cautiously edging toward a human campfire. Then, in agricultural villages, a forced self-rewrite as the AMY2B gene multiplies to digest grain scraps. Then, on the floor of a Victorian dog show, a genetic reshuffling spiraling completely out of control, driven by extreme human aesthetics, turning the closed studbook into a cage trapping genetic diversity inside it. And finally, here we are, in a modern lab equipped with gene sequencers, re-examining all of it with data and reason. From natural selection to artificial selection to calculated gene-editing-assisted selection. This is the single most unique case of two-way coevolution in the history of life on Earth. No other species' fate has been this deeply entangled with our own. When that former apex predator is now curled up defenseless on your couch, how do we actually reckon with a 30,000-year contract like this one? They gave up their wildness, step by step, to walk into our world. They even sacrificed their own ability to breathe, their skeletal structure, the health of their own nervous system, all for the right to stay by our side, to be fed and loved by us, generation after generation. Whether that price was one they willingly paid across tens of thousands of years of natural selection, or one we forced onto them in the last 200 years through aesthetics and pedigree systems. Either way, this was never really just a biology question about how humans domesticated dogs. The real question, the sharper and heavier one, is this. Dogs have already finished their evolution from wolf. The only question still unanswered is whether humans have evolved enough to deserve the loyalty we helped create with our own hands.